And oh,
No, keep going.
okay. And David gave here a talk, I think like a few weeks ago. And his talk focused more on the theory behind the origins of life and origins of evolution. And I'm an experimentalist. So today I'll be talking more about how we can experimentally approach understanding origins of evolution. And okay. So this is a note that I found at a desk of one of our undergraduate students that, in my opinion, perfectly exemplifies what the origins of life research is about. And this is because it basically splits the whole field into three main sets of questions. First is, how were the building blocks of life produced? So what were the sources of amino acids, nucleotides, lipids, and other biologically relevant molecules on early Earth that didn't require any biosynthesis or actual biology? And for that, we actually surprisingly, well, there's no clear consensus. We still surprisingly have a relatively decent understanding of what was happening. There are many known sources of organics like amino acids or nucleotides on early Earth, for example, from meteorite deliveries or hydrothermal activity and various other environmental processes. And then on the other side of things, we have the question of how did early life evolve once we already have cells, once we already have some kind of complex structures. And there's also a significant amount of research on that subject, mostly focusing on what we call a top to bottom approach, where, for example, we can look at modern microbes and from them try to deduce what their last common ancestor looked like and functioned. But then in the middle, we have kind of a big gray area where there is a question of how did we get from very simple chemicals to something that resembles life. And there is probably less consensus in that part of astrobiology and origins of life than in the other two. Because we know that at some point there were genes, at some point there emerged genetic inheritance, most likely in the form of RNA molecules, possibly with catalytic activity. This is the basis of what is known as the RNA world hypothesis. That is probably one of the most well-known origins of life scenarios, or at least parts of origins of life scenarios. But the problem is that even something like RNA is extremely complex, and getting a ribozyme and catalytic RNA molecule that is capable to replicate and do catalyze all sorts of complex reactions is very difficult to happen spontaneously. So there was likely something else before, and this is the part where it's not particularly clear what was going on Earth right before life actually emerged. And this is also what basically my research and my group's research is about. And this is also what I will be talking about today. So before we move on, it is important to talk about what is life. So biologists, I think, don't often think about this, but life is extremely difficult to define. And there is no consensus here at all, from what I understand, at least. And some people might even argue that there's no point of defining life, but we need to have some kind of a basis to understand the origins of life on Earth, and what specifically we're looking for when we're searching for life elsewhere, which is another aspect of astrobiology. And one of the most widely used definitions is what is known as the NASA definition of life, which is basically that saying that life is a self-sustaining chemical system capable of Darwinian evolution, where Darwinian evolution is basically just a response to selection to try to be agnostic to any kind of genetics that must have come later. And there are basically two important parts to this definition. First, we have Darwinian evolution, of course. So when we're looking for first systems that were lifelike, we're looking for something that was capable of evolution, of responding to natural selection. And another part is the word self-sustaining, which might sound somewhat vague, potentially intentionally, but what it actually means is autocatalytic. And this is a term that David Baum definitely talked about in his talk, but I'll revisit it here as well. Because autocatalysis is actually pretty fundamental to life today and to the study of origins of life itself. So autocatalysis, or autocatalytic cycles to be specific, are basically sets of chemical reactions that always include this reaction or reactions of this type, where A plus F goes to 2A, and A is just a member species, so any kind of compound, and what we call a member species. And F is a food compound, so a compound or a set of compounds that is consistently produced by the environment. So for example, constantly being pumped out by a hydrothermal vent or something like that. And something that is not particularly replenished inside the actual cycle. And also F cannot go to A, so this reaction is banned for the whole cycle to be autocatalytic, or at least the rate of the conversion of F to A should be low enough to be negligible. And this reaction might seem complicated, but everyone here is actually very familiar with at least one example, which is life itself, because every life cycle of an organism is autocatalytic. Like, for example, you have an adult, an organism, it lays an egg, that egg hatches into another organism, and that organism that hatched the egg still remains. So you have two organisms for the price of one, with the constant inflow of food and production of waste. So basically every single life cycle of every single organism is autocatalytic. And this is why autocatalysis specifically is very important to life. But the thing is that these kinds of processes are also known in chemistry, and we can start higher by looking at modern biochemistry. There are many biochemical pathways that are autocatalytic on their own, even without talking about the cell itself or the organism itself. Like, for example, if you run the Krebs cycle, a well-known metabolic pathway, in reverse, then it actually is autocatalytic because it produces 2 molecules of oxaloacetate per round, per one circle of the cycle. And into the context of origins of life, there are also a lot more, much simpler autocatalytic cycles that we know of. Like, for example, the Formose reaction or the Formose cycle, which is probably one of the most widely studied in the context of origins of life. And it looks basically like this, where it's just a cycle of a bunch of different ketones and aldehydes of varying complexity. And the member species here is glycoaldehyde, which is a 2-carbon sugar. And as you can see in the cycle, it's produced twice. So it's basically, if you remember, A plus F goes to 2A. So A here is the glycoaldehyde. And the food species is formaldehyde, which is a 1-carbon aldehyde, extremely simple, available in early Earth through a variety of means. And to start the cycle, all you need is basically just some glycoaldehyde, some formaldehyde, and then the cycle is started and keeps running. And as a result, you can get 3, 4-carbon sugars, and there are also a lot of add-on reactions that you can add to the cycle that will make the products even more complex. So coming back to the definition, we've talked about what self-sustaining is, and we'll come back to autocatalysis and specifically the Formose cycle too later in my talk. Now let's talk about evolution, because let's say we have a chemical system in mind and we're trying to show that it is lifelike or it is relevant to origins of life, and we show that it is autocatalytic, but then so that we can show that it is capable of evolution, we should be able to demonstrate that it has some form of heritability, either genetic, non-genetic, depending on what we're looking for, and that there is some kind of a contingency between different generations of this autocatalytic replication. Once we show that, we can then test whether the system can respond to selection, whether there can be some kind of a deterministic response to selected pressures, because natural selection is a deterministic process. And once we have that, we can claim something cool and maybe get a Nobel Prize. I don't know. But this kind of framework is basically what I use in my research to try to see if we can identify the simplest possible chemical systems that are capable of responding to natural selection. Because once we have that, we have some kind of a deterministic pathway to more complex things like we see in modern life. So I kind of split this talk into 2 parts based on the different systems that I will be talking about. So I'll start with lipid vesicles and then move on to more complex chemical ecosystems. Just to basically try to answer the question of whether autocatalytic systems can show heritability and evolution-like dynamics. So let's start with heritability in vesicles. So lipid vesicles are basically the most obvious candidate for some kind of a prebiotically plausible compartment. So a compartment that would have been available before life first emerged. And vesicles are composed of molecules that we call amphiphiles, that are basically molecules like this that have two parts to them. One is the hydrophobic tail, which is just a set of atoms that orient themselves away from water. And then hydrophilic head groups that orient themselves towards water spontaneously. And there are many different examples of amphiphiles, like in modern life, we have phospholipids or fats. And there are also a lot of examples of molecules that are much simpler and more prebiotically plausible, which just means more likely to be available on early Earths. Like, for example, a lot of different 10-carbon molecules like the carboxylic acids, amines, alcohols, and so on. And those molecules can spontaneously organize in aqueous solutions, so in solutions that have water, into either micelles, which are these structures that are basically a monolayer, a single layer of amphiphiles that doesn't, and they don't have like a hole in the middle, there's no water in the middle. Or vesicles, which are a lipid bilayer that allows for water and like all the water soluble compounds to be concentrated inside. And vesicles have a membrane on their surface, of course. And this is what is very similar between vesicles and cells, because cells are basically an instance of vesicles, because all modern cells have some form of plasma membrane and some kind of an aqueous compartment on the inside. And the similarity doesn't end there, actually, because vesicles, just as cells, are autocatalytic. So let's say we take a cell with its complex biosynthetic machinery, various enzymes, produces amphiphiles, so phospholipids in this case. Those amphiphiles get incorporated into the membrane and the cell grows. And then once it reaches a critical point in its life cycle, the cell divides. Meanwhile, if you have a vesicle, then it is constantly accreting amphiphiles from the environment, assuming they're constantly produced. And of course, you can imagine a hypothetical situation where you would have some kind of an autocatalytic cycle inside the vesicle that would make those amphiphiles, but it's still the same thing. And it is secreting amphiphials, it's growing, and it's yet again dividing once it reaches a certain critical volume. And this has been experimentally shown. And it also means that the processes by which vesicles behave and modern cells are actually fundamentally similar, because they're both essentially an example of the same reaction that I've shown before, where A is a vesicle or a cell, and F is just a food compound, in this case, amphiphiles. And this is what makes fatty acid vesicles very relevant to the study of origins of life. And it's also really interesting that depending on their composition, they divide at different rates as well. So like phospholipid vesicles are less likely to show spontaneous division than fatty acid ones, for example. Vesicles are also affected by their immediate chemical environments. So this is just a brief experiment that I did where I used a bunch of different prebiotic soups. And prebiotic soup is, by the way, an actual scientific term that typically means a mixture of compounds that are designed to approximate a certain set of conditions on early Earth, or conditions anywhere else as well. And I prepared vesicles in different soups, different mixtures of compounds, and measured their size. So on the x-axis, you see the radius of the vesicle, and the y-axis is just basically the proportion of how many vesicles of a given size there are. And the point is, you can see that the size distributions of these vesicles are vastly different depending on what solution they're in. Whether they're in a solution that is based on iron meteorites or carbonaceous chondrite meteorites, or just in a rich soup that kind of has everything, and we'll get back to it in a bit, or water. And this is because there are also a lot of other different compounds that are not amphiphiles that can interact with vesicle membranes and produce some interesting phenomena. Like, for example, amino acids and nucleobases have been shown to bind to the membranes and stabilize them against various environmental pressures, while something like high salt concentration can, in return, disrupt vesicle formation and cause constant leakage. But in addition to all of this, there are also some really interesting phenomena that were shown before related to fatty acid vesicles that are actually directly connected to their potential for evolution. First of all, it's the heritability of the size of vesicles themselves, which is exemplified by a phenomenon called the matrix effect, where basically if you mix monomers with vesicles of a preformed size, you get the same size distribution of vesicles repeat over and over. So for example, let's say we have this template solution of vesicles where x-axis is again the radius and y-axis is basically the proportion. And you can see that the distribution is super narrow. But then when you mix this with a much more heterogeneous sample that has micelles, that has crystals, that has monomers, that has a whole bunch of things that are not necessarily vesicles. And if you mix them at any ratio, by the way, you can have 1,000 of A and 1 of B, and you will still have the same result as the template itself. So the distribution will still stay narrow. And this can be somewhat equivalented to what is known in evolutionary biology as robustness, where there are some mechanisms in organisms that would intentionally decrease the variation. Like, for example, DNA repair is one of the most obvious examples in almost all cells, where it tends to make the whole population, decrease the variation in the whole population by preventing additional mutations. And this is kind of the same here, at least fundamentally, but for vesicle size specifically. And another interesting phenomenon that was studied more so computationally, I don't think there were any experimental studies done on this before, is compositional inheritance, which is basically a model of inheritance that is analog rather than digital, like we see in DNA today, where a parameter that is actually inherited is just the proportional composition of the membrane. So let's say you have a vesicle composed of many different types of amphiphils, all at different proportions, then you can have the inheritance of n mutations within that proportion of amphiphils inside the membrane, or even some molecules in the amount of some molecules that are associated with the membrane itself that are not amphiphilic. And this is basically analog because it's based on proportions, versus in DNA, you have digital inheritance because you have nucleotides coding for amino acids and so on. And this has been shown computationally for both vesicles and micelles, like shown here. There's actually a lot more studies on micelles these days, where you can have various compositional mutations happening and actually being consistently inherited across multiple generations of replication. So for my studies, I was trying to develop some kind of an experimental framework to just broadly look for heritability or heritability-like patterns in fatty acid vesicles experimentally. And I use what we call recursive seeding, where we basically have a fresh vesicle solution that we just prepared, and then we incubate it for 24 hours in this case, and this will be our generation 0. And then after 24 hours, we make another fresh vesicle solution, and we mix the new fresh solution that we prepared with the incubated one at a certain ratio. Like for this experiment, it was 9:1. So we took 90% of the new fresh solution, mixed it with 10% of the incubated solution from generation 0. And then we incubate the resulting solution again for 24 hours. And at the same time, we also incubate the fresh solution without any transfer from the previous generation. And we call it no transfer control, or NTC, because there were no transfers, and we make them also every single generation to compare with the transfer samples that we do for surgery generations, and also to control for any sort of experimental variation that results from us just preparing the solutions differently on a given day. And as a result, we want to compare the various vesicle properties between transfers and NTCs in every generation and see what kind of pattern we observe. So for actual solutions, we chose 3 amphiphils mixed together. We have decanoic acid and decylamine as our main vesicle forming molecules, which are just 10 carbon, carboxylic acid, and an amine. And we also included the detergent, Trident X100, which is not particularly prebiotically plausible, So it's definitely not around in early Earth. But we added it still because it introduces additional heterogeneity to our size distributions and a bunch of other parameters of our system. And it also is known to promote vesicle division by just inducing binding in the membranes. So this is why we kept it there, because we do want our system to be more heterogeneous for the purposes of the experiment. And as our solvents, so the solutions in which we actually prepare those vesicles, we either use water or what we call enriched prebiotic soup, which I mentioned before, which is just a mixture of various compounds that were likely present in our early Earths, very loosely, because there are a total of 70 compounds here, stuff like amino acids, nucleobases, sugars, and so on, and a bunch of salts too. And our goal was to, again, compare controls with transfer samples every generation in this experiment. And we have a pretty clear set of expectations for what we would expect of some kind of a vesicle property that we measure, where no transfer controls should be identical every generation, because they're supposed to be, at least, unless there is some kind of a experimental error or unpredictable indeterminism within our system itself. Then our null expectations for the transfer samples is actually that they will increase according that the value of that certain property will increase or decrease according to our transfer rate. So let's say we have a certain property that changes over the course of an incubation. And then when we mix 10% of the incubated solution with the fresh solution, then we expect that property to increase by 10%, because 10% is the amount we transferred. And then in the next generation, it will increase by 1%, and then 0.1%, and then so on. So this is our null expectation that wouldn't really tell us anything. It wouldn't necessarily say us that we don't see anything interesting, but it wouldn't necessarily tell us that we do. But then if we see some kind of a nonlinear dynamic where we see some form of autocorrelation in our between different generations, then that is what we would describe as a positive result and likely presence of heritability in our system. And there are a lot of different dynamics that this can take. Like, for example, oscillations are actually probably some of the most interesting things we can observe, because oscillations are actually really common in many autocatalytic systems and life itself. Like, for example, predator-prey dynamics, like the famous Lotka-Volterra model, which is just a super simple set of equations. And this is happening in life as well, where you can see basically phased oscillations of predator and prey population over time. And something like this was also shown in simple autocatalytic systems by some members of our lab in the past. So seeing something like that would actually be extremely interesting, and it would allow us to understand a little bit more about our system. And now, coming back to the expectation plot, remember that y-axis, it was vesicle property. So there were a few things that we actually measured in this experiment to try to see what kind of dynamic we observe. The first one is fluorescence of now red. And now red is this fluorescent probe that is actually not red at all. It's beautifully purple. But now red fluoresces exclusively in hydrophobic environments, because the molecule itself does a twist, and there's a charge transfer happening between different parts of the molecule that allows for fluorescence. And because it's fluorescing only when the molecule is twisted, the intensity of its fluorescence is actually dependent on what kind of hydrophobic environment it is in. Let's say it's a particle, then like a vesicle or micelle that has those hydrophobic tails either on the inside or inside the bilayer, then the fluorescence will be proportional to the area to volume ratio of the particle itself. So in something like a micelle, now red will fluoresce more because area to volume ratio is greater. And in something like a bilayer, where the molecule is deeper inside, it will fluoresce less because the area to volume ratio is smaller. So this is a kind of a very broad experimental parameter, because there are a lot of things in which area to volume ratio depends. Like, you can have vesicles of different size, you can have some fraction of micelles in your system, you can have like different number of membranes stacked on top of each other. So, and this is actually good, because that way we're able to kind of catch a lot of different things that might change in our experiments all at once. And surprisingly, we actually see oscillations in the fluorescence of Nile red over the course of 30 generations of this experiment. And even more surprisingly, we see oscillations in both samples with water and EPS, the soup. So the y-axis here is just the ratio of fluorescence beam transfers and controls, and stars just mark generations in which this difference is significant. But basically, we can see that there are 11 generations in which the difference is significant for water samples, and 6 generations for EPS. So this is even more surprising. But also, this means that it's very unlikely to be noise, just because it's 6 and 11 out of 25 total measured generations. So this is very interesting, and we decided to explore this pattern further by calculating the autocorrelation function, which basically just tells you if samples, once you have a time series, you can calculate correlation between values different by a certain step, and the step is known as lag time. That's on the x-axis of this plot. So it's basically a period between which period in oscillations between which correlation is observed. And we calculated the autocorrelation function, and values that are greater than greater or lower outside the range of the confidence interval we would consider significant. And as you can see for EPS samples, when we calculate autocorrelation for ratios of transfers to NTCs that I've shown on the previous plot, we actually see significant positive autocorrelation for EPS at lag times of 1, 2, and 4. And for water, we see significant negative autocorrelation for lag times of 2 and 5, which is really interesting. And it's really interesting that they're also facing the opposite directions. But then we actually calculated the same parameter for both transfer and NTC values individually. And unfortunately, we also see that NTCs do oscillate somewhat over time. In both water and EPS, we see significant autocorrelation in NTCs, and obviously in transfers too, which is also greater. So it's still likely that we observe the effect. It's just that either our system is too indeterministic, or we messed something up that we're still not sure what. But there's definitely some variation in controls. So in conclusion, we can say that there's likely some kind of a heritability of this parameter in our system, but the exact pattern and period is very difficult to determine, especially in EPS, because the autocorrelation in NTCs is so great. So yeah, and we think it might be, we hope it is, because of our system is indeterministic, because Triton does add quite a lot of variability to various parameters that might impact fluorescence of Nile red. But we'll see. We need to do more experiments on that. In addition to Nile red, we also looked at a bunch of other parameters, like we measured vesicle size using a method known as dynamic light scattering, where basically you shine a laser at a solution of particles of given sizes, and those particles are diffusing in the solution, and larger particles will diffuse slower, so they'll have more time to pass through the laser, and therefore scatter more of the laser light. And that way you can actually determine pretty accurately the distribution of sizes, distribution of radii of those particles in your solution. So again, x-axis is the radius here, y-axis is basically the proportion, the intensity of light scattering. And we can see differences between transfers and controls in almost every generation that also seem to not be following a clear linear pattern. So in both water and EPS. So there might be oscillations there too, but we don't have enough data to actually determine that, because in this case, it's very difficult to develop one kind of index to compare across all generations, since we're looking at a whole distribution within the solution. And also each generation takes like 8 hours, so we didn't analyze all of them. But there might be some kind of a interesting pattern too that might also align with the Nile red oscillations. But what is perhaps the most surprising is that when we looked at chemical composition of EPS over multiple generations, we actually see no difference between transfers and controls at all. So these are principal component plots based on data from LCMS, liquid chromatography, mass spectrometry tool that I'll also come back to in the second part. And you see that we have basically no separation between transfers and controls at all in generations 25 and 30. And this is very surprising because, for example, most of the reason we looked at this in the first place is because we were trying to see if we can see any kind of pattern of compositional inheritance in our system, because typically, like most computational models of compositional inheritance, like the famous lipid world paper, they base their models on systems that have not only heterogeneous membranes, heterogeneously composed membranes, but also heterogeneous solutions, heterogeneous insides of those membranes. So where you can have a bunch of different compounds that tend to preferentially associate with different types of membranes and potentially even catalyze some important reactions. And if those kinds of processes were responsible for the differences we see in our now red data, where we see oscillations, then we would likely see at least some kind of a difference in the chemical composition of EPS, and we don't, which is really surprising. And to sum up this part of the talk, basically we observe three things: oscillations in one of the parameters, now red fluorescence, which might imply some kind of nonlinearity in either vesicle size, micelle fraction, or vesicle lamellarity, which are the parameters that change according to area to volume ratio. We do see deviations in size between transient controls that implies that there is an effective transfer history on that parameter, at least. And somehow we see very few changes in actual chemical composition of our samples that have EPS. That implies that whatever patterns we're seeing in other parameters, it's very likely that it's either based on relatively few chemical species that we might just either not be able to detect or just miss with our analyses, or is based entirely on the physical properties of our system. So to answer the question of whether vesicle populations can show heritability, we can very cautiously say yes and keep doing experiments, which is what I'm doing. So now I want to transition to the next part of my talk, which will be more about that part that I talked about earlier about compositionality, because here I'll be focusing on much more complex chemical ecosystems and trying to see if I can also see some kind of a nonlinear dynamics in them. So let's say we take this very simple set of chemicals. We have three organic molecules, acetate, formate, and methanol, 3 different salts, and a mineral, pyrite. And we do basically apply basically the same experimental design to that solution as we did to vesicles and EPS or water, where we would also take a fresh solution, incubate, transfer 20% in this case, then incubate the fresh solution again, and just keep going for 10 generations. And with a few differences, right? Here we have a mineral phase, we have pyrite, and we also autoclave at the beginning of each generation to prevent any kind of biological contamination, which would basically ruin the experiment. But instead of focusing on physical traits like vesicle size or lamellarity like we did in the last experiment, here we'll be looking specifically at the composition using LCMS, using liquid chromatography mass spectrometry. And since we have such a simple set of compounds that we call the food set, it will be a lot easier to analyze its composition. Whereas if we had a population of individual vesicles, it would be much more difficult to understand the composition of each individual vesicle. And here we don't actually know if we have autocatalysis in the solution. This is just a bunch of very simple molecules that are definitely present in our layers. But since we are autoclaving and we have a mineral with a potentially catalytic activity, we will likely have a lot more compounds in the end of the experiment. And there are some expectations that we can think about. So first of all, if we actually do observe autocatalysis, then in an ideal situation, if you have this kind of reaction with a reversible reaction as well, reverse reaction as well, then you would expect some kind of a logistic growth in the concentration of A, because it will increase and then it will plateau and it reaches equilibrium with the reverse reaction. But this is a very idealized situation when you basically only have A and only have F in your solutions, and F is not producing A on its own. But in reality, you're very likely to have, especially since we have a lot of different chemical species present in our solutions, it's very likely that there could be other reactions, like some reaction that will consume A and produce waste, or another competing autocatalytic reaction that will compete for F and will therefore might generate some kind of oscillations. So basically, the more reactions and the more chemical species you have in your system, the more complex dynamics you're likely to expect. And this kind of dynamic can actually get pretty complex, even for relatively simple chemistry. Like, for example, the Belousov-Zhabotinsky reaction, which is a pretty well-known example of autocatalytic chemistry, actually produces oscillations that are spatial if you add a certain indicator. So you can actually mix your reagents in a petri dish and see something like this, which is super cool and also a great experiment to like demonstrate what kind of dynamic we're expecting. So as I mentioned, we have, and for our system, we're using autoclaving and we add pyrite. And both of those things result in what is known as combinatorial explosions, where you start with a relatively small set of compounds, but then once you autoclave in the presence of pyrite, you get a lot more compounds added to your solution. And you can see that actually, if you look at the number of new compounds, number of enriched compounds, then pyrite itself actually has an even greater effect than autoclaving, which is basically autoclaving is basically heating to 120 degrees Celsius and under high pressure. And pyrite has an even stronger effect, significantly stronger than autoclaving. And when you combine both, you get an even greater enrichment in compounds. And pyrite is this really nice cube-shaped mineral that is kind of grayish in color and is composed of iron sulfide, and is actually a very prebiotically relevant mineral because it easily oxidizes in today's conditions because we have a lot of oxygen in our atmosphere now. But back in the Hadean, back 4 billion years ago, there wasn't any oxygen in the atmosphere. So there was likely a lot more pyrite available on Earth's surface. So in addition to seeing this explosion over just the course of one incubation of our food set, we also do see that transfers very rapidly enrich for a lot of different compounds. Like this is another PCA plot where you can see a very clear separation between controls and transfers. And this is the plot of generation 10, but we see this in basically every single generation. So essentially, now we want to see if we see some kind of a taming of this combinatorial explosion that we observed over the incubation time. So whether we can determine if there is some kind of a pattern in chemical diversity over time. And when we look at the percent of total compounds that are enriched or depleted in transfers compared to controls in our solutions over the course of multiple generations, we first see that it increases in the first few generations. And you'd think that it would keep increasing, but actually no, it decreases all the way down to almost 0 in generation 6. And then you would expect that it would stay the same and like plateau at least somewhere. And it doesn't, because it yet again increases. And this is really cool. And it's actually a shame that we did this experiment only for 10 generations, because it would be great to see how far this dynamic can go. And if it would actually plateau at any point. But then we also measured a bunch of other statistical parameters, like we calculated the Bray-Curtis dissimilarity index, where we looked at basically calculated how different are transfers from controls and how different are different samples of transfers between each other and different samples of controls between each other. And you can see that the difference between transfers and controls is always greater than the intra-control or intra-transfer difference. And the dynamic here is also nonlinear, also kind of resembling the pattern that we see when we just calculate the percent of enriched compounds. And we also, one of the cool things here is that since we are talking about basically a chemical ecosystem, we can apply the same techniques that ecologists use to analyze biodiversity. Because in our case, instead of actual organismal species, we just have different chemicals. But with LCMS, we also get basically the list of all of those compounds and their relative abundances, which is kind of similar to a data set coming from an ecologist. And we can calculate various parameters like the alpha diversity, like the Shannon alpha diversity index, which just shows the diversity within the group. And this is just an example. This pattern is very unclear, and I don't actually understand what it means. But we do see a significant difference between transfers and controls. Several generations is just different from what we would expect. But this is also a direction we're trying to explore more now, just trying to develop a more ecology-based framework to look at our data. But another great thing about LCMS is that we can also focus on individual compounds and try to see if we can actually explain any of the patterns that we observe, which is much easier to do than if we had, for example, vesicles, where a lot of physical parameters are involved. So we actually detect glycolaldehyde in basically everywhere in this experiment. And if you remember from like one of my earlier slides, glycolaldehyde is the autocatalytic member species of the Formose cycle, of the Formose reaction, which is one of the most well-known prebiotically relevant autocatalytic cycles. And we see some kind of nonlinearity or just incubation time. If you just have the food set incubated for 24 hours, it does seem like there's some kind of a difference from t equals 0, at least in 14 hours and 22 hours. Then in 20% transfers, we don't really see much enrichment at all, except in generation 7. But then in 50, when we repeated that same experiment, but we changed the transfer dilution rate from 20% to 50%, we actually see that glycolaldehyde is enriched in transfers every generation, at least every generation we analyze, which is so far only 4 out of 15, which indicates that it's pretty likely, and just the fact that we are detecting glycolaldehyde that we didn't add in the first place, indicates that we very likely do have Formose cycle just randomly happening in our solutions. And here's just a reminder of what the cycle looks like. We don't really detect many of the other, we detect a bunch of other aldehydes as well and ketones that are still trying to piece together the whole reaction network that is actually happening. But what we think is happening based on some other tests is that since we have formic acid in the food set itself, we think that it gets reduced to formaldehyde, either with the help of pyrite and autoclaving, and then formaldehyde dimerizes to glycolaldehyde. And once you have both formaldehyde and glycolaldehyde in your solutions, you basically have the whole cycle running because you have A and F present. And if you remember, A plus F goes to A, you need to have A in order for the cycle to actually happen. But then we also found a bunch of other species that are really interesting and also likely are part of autocatalytic cores that are present. So one of them is hexylamine, which is a pretty simple 6-carbon amine that very clearly oscillates. It's significantly enriched in transfers in every generation except 1 and 6 in the 20% transfer experiment. And if you remember the previous plot that I've shown about the percent transfers, then you can see that the profile of the percent of total compounds that are enriched and the profile of hexylamine abundance is actually nearly identical, which is really exciting because I've just found this randomly by like scrolling through folders of plots. And it seems that it is likely that hexylamine is either the cause or the consequence of whatever complex processes we have in our solutions. And I mentioned that this isn't necessarily the proof that hexylamine is autocatalytic in any way. Plus, over incubation time, we also see oscillations. However, when in the repeat experiment with 50% transfers, we actually see an increase in plateau, which is basically a dynamic we would expect from an autocatalytic member species. So this does indicate that is possibly another component of a certain autocatalytic cycle present in our solutions. And in addition to that, we also see a bunch of other compounds that are likely chemically related, like hexene sulfonic acid, for example, that also show a pattern that is somewhat similar to hexylamine. And what we're doing right now, actually, is trying to see if we can find any compounds with a pattern that is reverse of what we see in hexylamine. Because if hexylamine is indeed an autocatalytic species, then the food set for that autocatalytic reaction should have the inverse pattern. Or at least, even if not perfectly inverse, it should be depleted in a lot of generations compared to controls. And we did find a few candidates for that too. So to summarize this experiment, we see pretty clear nonlinearity resulting from very simple solutions. Again, like all we had for this experiment, all we started with for this experiment is basically just 6 compounds, 3 organic compounds, 3 salts, and a mineral. And as a result, we get pretty clear nonlinearity. And also, one thing that I forgot to mention is that when we have this kind of recursive seeding framework, when we have a fresh solution, mix it with incubated solution, this is a very simple, it's very practically applicable because it's just very simple to do experimentally. But it also does kind of represent a bunch of early Earth environments. Like if you think of a tidal pool, then it gets mixed with the ocean at some point, and then the ocean comes out, the ocean leaves, and the pond is drying out. So you basically have the same kind of a transfer happening like we have in our experimental framework. And this is applicable to both vesicles and this experiment. And we also possibly see emergence of autocatalysis. And we're still trying to reconstruct all the reaction networks and figure out some better ways to analyze this data. So this is more of a work in progress. But yeah, it's very exciting that we see some pretty interesting results here. So to come back to this beautiful figure, I want to try to give a somewhat of a framework of how we can look, just to summarize how we can look for origins of life, or at least origins of first living like organisms. So at some point, we have the emergence of autocatalysis. Then we have heritability. We have an autocatalytic system that is capable of transitioning between different states with some form of historical contingency. And then we have response to selection, so evolution. And then once we have a system that, a simple chemical system that can respond to selective pressures, so it can evolve deterministically, then we have a path to increasing complexity. Although it is important to note here that evolution does not always result in increasing complexity. That's literally not always the case. But it is still a mechanism to get there at some point. And then we get to genetics, then we get to cells and first life. So I went to sync my lab group, all the undergraduate and graduate students who helped with this project. And yeah, that's it.
Plenty of time for questions. Please go ahead.
Yeah, so have you looked at constructing any molecular identity networks to see how, like, you show the exponential change over time? But have you looked at any molecular identity networks?
No, not yet.
Because that could show, like, because you assign like you have fragmentation data for all this stuff, and so you have structures to assign cosine scores to see how similar compounds are, and then I would suggest looking at those, because then maybe over time you'd be able to visualize, oh, this compound group like raises in intensity, and this one decreases, and that might be a lot more efficient. I think like compound by compound and seeing like who's changed more. It can show relationships better that might point towards these valid cycles.
Yeah, so we didn't do that yet because the thing is that in our data sets, right, we use with the analytical pipeline that we use for LCMS data, we get a lot of compounds that don't have a name or a formula and are just rows in a spreadsheet. And so far we try to still include them for many of the analyses, and maybe at some point develop some way to like analyze them better, because what we're doing, like our methodology is basically what is known as untargeted metabolomics, where we don't have any specific, when we're starting our experiment, we don't have any specific compounds in mind. We're just trying to catch as many things as we can. Yeah, so we haven't tried that yet, but I am actually in the process of trying to figure out how to write code to convert all of this data into formats that would work for this, because you basically need like Smiles IDs, which is just like ways of writing up formulas. Yeah, definitely. But we don't look at like individual each compound by compound though. Really, we like generally just scan. Like one of the things that I did with this data is just scan for correlations. So like look for the compounds that have significant correlation with hexylamine, for example, and then look at their dynamics individually. But those that do have a significant correlation, try to see if they can be like connected chemically in any way or like make sense chemically.
Cool.
Great talk. I have a couple of questions. First one is related to the first part of the like studies. So maybe you can answer it by rolling, well, now going forward.
Forward.
Couple slides. Okay, so how do you know, like, whether the second pass on your chemical system, on the diversity of your chemical system is with, like, going back to the same place where it was, or is visiting like a new state, but it's like there's like a stepwise evolution of the system?
So you mean when you're, so how do I know if the state is the same, or so basically how define the boundaries of a state?
Yeah, well, I can imagine that like there's like you can increase the diversity of the number of compounds you have in the second class, but maybe those compounds.
Oh yeah.
I think that maybe you have
Yeah, I mean, we calculated a lot of different things for this because it's pretty new and there are a lot of different techniques that basically say kind of the same thing. So we calculated the dissimilarity index for basically each compound. So it's still is taking into account like the specific compounds that are different. But yeah, when we calculate something like diversity, it is just pretty general. It could be different compounds as well. So there is also another technique that we did that I haven't shown here, where you can do the supervised analysis, like OPLS-DA, the ordinary partial least squares discriminant analysis, where as a result you can get basically the importance. If you see like some kind of a separation between, let's say, transitive controls, then you can determine like which compounds are most responsible for that separation, which is something that is often used in chemical ecology, I think, in something like plants or animals. And that way you can compare like which compounds are actually, if the compounds are the same across generations. And many of them are. So it does seem like when we see the same state, it's likely caused by the same-ish phenomena. At least in the second part. In the was now red, it's a lot more complex. But
yeah, I also have a question for the first part.
Mhm.
In this case, I understand like the systems holding, they're like changes in the size and actual practice, etc. And I was wondering what if these like driving these changes, even though money like the vesicles don't have agency there. So like the whole thing might be moved by vision.
Mhm.
So in that case, what would be like
yeah, so I'm not exactly sure about that. I was hoping that LCMS would give some kind of light to that, because I think that it is likely the third part, as in there are some few chemical species that we just don't detect. Because the thing is, I mean, there are reactions happening to our amphiphals in the solutions. We only had 3, but we never actually ran LCMS on samples with Triton. This data is for samples without Triton, because Triton is very LCMS incompatible. But basically, they're like deoxynitric acid and decylamine could oxidize. You can get like a bunch of other reactions happening. You can get diacetyl amino acids forming because we have a bunch of amino acids in EPS. So you can have some kind of a diversity in chemical composition. And you might have, let's say, the total concentration. Let's say you have like 3 amphiphals in the system. You might have their total concentration remain the same, but their distribution within vesicles can change. So like earlier at first, we might have like vesicles was just 33% of each of the 3 amphiphals. And then you might have like vesicles that are 100% of each of the 3. And this can happen because of like different reactions happening, or like new reactions that were selected for, or some other changes, like changes in physical properties like size and so on, or even fraction of micelles too. Because, but this is all pure speculation because we haven't done any actual tests to see that. And a way to do that would be to use something like TOF-SIMS or like microscopy, electron microscopy. Because fun fact, I've been doing these kinds of experiments for like over a year, and I've never actually seen these vesicles. Because electron microscopy is pretty expensive, and I've kind of been mostly focusing on other techniques. And I've just been detecting them using DLS, using size distributions. And this is also like another thing too, like in this, based from size distributions, I didn't really talk about it, but we can't come up with a clear explanation for null red anyways, because in some samples, we do see a greater fraction of what are likely micelles, because micelles are much smaller. Micelles are, for pure trident micelles are about 10 nanometers. So they're like right here. And you can see in some generations, like generation 11, we do see a greater fraction of micelles that does actually correspond to a much greater fluorescence. But then in others, you might see a major difference in size, but you don't see any difference in fluorescence, or you see a big difference in size, and it is correlated to fluorescence in the right way. And it does seem like there are other parameters at play that we just didn't measure. There's also similarity too, which we did measure, and it could potentially explain things, but it will also be reflected in size anyways, because vesicles with more lamellae, with more layers of membranes, basically will be larger.
Yep. So you observe a greater or lesser convergence between transfer and non-transfer. What are the implications for that for incipient heritability of the system? Would you expect a more heritable system, autoheritable system to be more stable with respect to transfers or more divergent?
So this is a great question, and this is something that is kind of difficult to conceptualize when we're not talking about genetic systems, because like typically when we're talking about heritability, we just mean correlation between some kind of correlation between parent and offspring, and you can have, but then in organisms you do have mechanisms to just stay within a certain attractors, like stay within a certain range of values. Like I've talked about robustness, we have like DNA repair, we have chaperone proteins, stuff like that, which is still like technically part of the heritability mechanism, but it's not something we would consider heritability typically. But basically we are looking broadly for any dynamic that would not be something that we could just predict from simple steady state chemistry, because life itself is out of equilibrium. We are hoping to see some kind of an out of equilibrium dynamic where like most chemists would typically expect approach to equilibrium and staying at equilibrium. And even in organisms too, like something like natural selection, it can obviously like move the system to some kind of a steady state in which it would metastable state in which it would stay for a certain period of time, just because that is where it has a greater fitness at a given time. But then when environment changes again, it will move again, and so on. So we don't actually have a consistent expectation. It just has to be something that's not directly predictable by basically equilibrium chemistry.
There's some, I'm wondering about NASA's definition of life and why didn't they specify Darwinian evolution? That they rejected any neutral explanations?
So I'm actually not sure about that. I've also heard that this isn't actually the NASA's definition. I don't really know why people are saying that, even though it is on the NASA website. But basically, I think the reason why it's saying Darwinian is because they're trying to avoid necessarily linking everything to genetics. So I don't know if that's necessarily connected to neutral evolution specifically, but or at least that was in mind here when this definition came up. But I think in this case they just mean response to selection, because that's something that is much easier to detect in a non-genetic system. And this is also like defining life in general is very difficult, and you can also make a very solid argument that you don't need to do that in the first place, because whenever we try to define life, we always base ourselves, we try to make a predicted definition, right? So we can apply it to origins of life or search for life elsewhere. And then when we do that, we have to base ourselves in like the three assumptions that we have of archaea, bacteria, eukaryotes are alive, and then what else is. And this is kind of might be kind of a fruitless endeavor unless you are trying to develop some kind of a operational framework. So this is why this is called like the operational definition of life, the working definition of life, because this just gives a clear set of outlines for what to look. And it also might have been written by chemists who might not know a lot about evolutionary biology. I'm not sure. But yeah, that's what I'd say.
I think they're just trying to say heritability, like, so entities that are heritable are going to go just on natural selection.
Yeah.
Just means that you have a metabolism, you have heredity.
Yeah, basically, I mean, the one does follow from another.
But so you have to have selection. I mean, I'm not, in reality, of course, there's selection, but I don't know that we have to have that in origins.
Yeah, that's, I mean, the thing is that once you have some kind of a deterministic process like selection, then you have probably a better way to get from just a simple chemistry to something that's alive. So that's probably why there is a greater emphasis on selection compared to other processes. But yeah.
All right, well, we are out of time, unfortunately, so big hand for Tym Sokolskyi.
Thank you for coming.
It's really, really good.
Thanks.
So now, basically, undergrads
Stay here.
have their turn to ask you
Okay.
questions. But I do have to take